Okay, now we're going to move into chapter 6, which is going to cover the muscular system. Now, this is a large chapter, so about halfway through, I'm going to stop and start a new recording. So you'll actually have 2 recordings to listen to, and this way it's a good breaking point so you're not trying to get too much at one time. Again, like with the past chapters, there are objectives for you to go back and look at and make sure that at the end of this chapter, after you've listened and to both the recordings and you read the chapter, you should be able to go back and answer these questions easily. Alright, let's start off with muscle functions. Alright, the major thing is to produce movement. Most of this is due to skeletal muscle, like mobility, simply moving you around, body language, responses to environment. And there's also posture muscles, stabilizing joints. That's a really big function of muscles, and most people don't really think about that. And that's why you have people that exercise more typically have less injuries because those muscles are toned around the joints. Now, muscles also generate heat, and that's a byproduct of muscle activity. And think about that. You already knew every time you get up, you move around, those muscles working produce heat, and that's why you get hot. Now, they also use ATP for muscle contraction, which of course produces heat. Now, for muscle tissue types, there are 3 types of muscles. There is skeletal, cardiac, and smooth muscle. Some of the differences, well, they differ in structure, where they're found, and what stimulates them to contract. Some of the similarities, skeletal and smooth, are both elongated, and all 3 contract due to myofilaments. Alright, so for skeletal muscle, some of the characteristics, it is striated, which means stripes or striped. It's also voluntary, which means that you have to actively think about it for it to move. It's soft, fragile, but has powerful contractions. It's attached to the skeleton, and it tires easily, so you need to rest it. Smooth muscle is not striated, so it has no stripes, and it's involuntary, which means you don't have to actively think about it for it to work. Now, that makes sense when you find out where it's located. It's found in the walls of the hollow organs, which means your stomach, esophagus, intestines, all your blood vessels. So we don't actively think about moving food through our digestive tract, right? No, so it's involuntary. Now, it has a single nucleus, and it's arranged in sheets or layers. The contractions are slow and sustained, and it doesn't tire. All right, here's an illustration showing the intestines, and inside of it, you can see there are smooth muscle. Now, in the intestines, there's actually 2 layers of smooth muscle. One runs longitudinally, and one runs circularly. And what is it doing is it's actually moving the food through the intestinal tract by contracting. Cardiac muscle is striated, so the stripes are back, and it's involuntary. Again, meaning you don't have to actively think about it because we don't think about our heart beating. It does it all on its own. Now here you have branching cells that are joined by intercalated discs found only in the heart. It's cushioned by connective tissue and arranged in spiral bundles. It contracts at a steady rate, but can increase the rate. Now, you'll talk more about cardiac muscle when you get into the heart chapter. All right, so for the next period, we're going to talk about skeletal muscle. All right, so muscle fibers are actually the cells. So you hear those words interchangeably. But anyway, so you have these connective tissue coverings. Each fiber or cell is covered by the endomysium. And again, all that is a connective tissue covering. Then we take all those muscle cells and we group them into fascicles, which again you can see on the illustration to the right. Each fascicle is then covered with perimysium. Then we take a bunch of fascicles, group those together, and that makes up an entire muscle, which is then wrapped with epimysium. Now that epimysium, that outer layer, blends into tendons or aponeurosis. A tendon is cord-like, where an aponeurosis is more sheet-like. They are both connective tissue that connects muscle to bone and provide durability and help conserve space. All right, so let's look at the microscopic anatomy. You have the sarcolemma. That is the name of the plasma membrane of a muscle cell. Now, we do this a lot in anatomy. Why do we have to give it its own name? Well, if I'm talking to another medical professional and I want to talk about what's happening on the sarcolemma, I can simply say, on the sarcolemma, this and this happened. If I don't have its own name, then I've got to say, well, the plasma membrane, but only of the muscle cell, not the other cells. So you have to add all these extra words, and then you think about that. If you have to do that on everything, it gets ridiculous. So that's why we give you these specific names to remember. So sarcolemma is the plasma membrane of a muscle cell. Muscle cells are multinucleated, and they're kind of pushed aside by myofibrils, which are organelles. Now, the sarcoplasmic reticulum, that is a specialized type of endoplasmic reticulum, which is going to store calcium, which is vital for contraction. And it also wraps around the myofibrils, which again are organelles that also function in contraction. They're made of chains of sarcomeres. A sarcomere is the smallest contractile unit. And when skeletal muscle contracts, it contracts sarcomere by sarcomere by sarcomere. Now, these sarcomeres contain myofilaments, which are responsible for the striations. All right, so in this illustration, you can see one muscle cell or fiber. And then if you look inside, it's actually showing you, well, first of all, in the top picture, you can see where part of that sarcolemma has been pushed back, the cell membrane, and you're seeing all the organelles and the nuclei inside. We're taking one of those myofibril organelles and we're pulling it out, and then you can see at the bottom what it actually looks like. Okay, notice that it has that striped appearance. Those are the striations. And then you're showed one sarcomere. That's one contractile unit. All right. Those sarcomeres. Now, you have two major types of myofilaments found in the sarcomere, myosin and actin. Myosin is a thick filament, and the protein here, of course, is myosin. This requires ATP for proper contraction, and you have myosin heads. These are projections that link thick and thin filaments together, and I'll show you a picture of that in just a second. Actin is a thin filament, and here the protein is actin, and it's a regulatory protein. It's connected to Z disc, which is a membrane between the sarcomeres. All right, if we look at this illustration, again, you've got one sarcomere. Now, the thick filaments are the myosin filaments, and notice those heads, where actin is the thinner filament. And what's actually happening in the muscle contraction is those myosin actin are going to bind and start pulling. So that's where you get the contraction, but we'll get to that in a second. So which of the following muscles are not striated and involuntary? Again, you've got skeletal, smooth, cardiac, all the above. The answer would be B, smooth. The connective tissue covering a muscle fiber is called the, well, remember, muscle fiber is just a cell. So it's A, epimysium, B, paramysium, C, endomysium, or D, a tendon. The answer would be C, endomysium. Next, let's look at the properties of skeletal muscle. You have irritability. That means they can receive and respond to a stimulus. Contractility. They can shorten forcibly. Excitation, contraction, coupling. This is where you get a nerve impulse, which is going to be an action potential, and you're going to have sliding filaments, which are going to require calcium. All right, so in this picture, we can see a couple of things. Now, all this, I'm sure, looks really crazy to you right now because you're like, what the heck is all this? But this is actually showing you a little bit of skeletal muscle contraction. In the upper left-hand corner, you're going to see the axon, which is part of a nerve. And then you see an arrow coming down, and then it shows that sarcoplasmic reticulum, which we just talked about, storing calcium. Now again, let's kind of get more into the skeletal muscle contraction, and then we'll come back to pictures. Okay, now you start off, what is a motor unit? A motor unit is one neuron and all of the muscle cells that it stimulates. There are small motor units and large motor units. Small motor units would have one neuron and just a couple of cells. So where would you see this? Well, around your eye muscles. Okay, the reason they have small motor units is because they are very precise movements, that sometimes you're only moving a couple of millimeters. Large motor units would have one neuron and hundreds and thousands of cells that it's stimulating. So those would be like your calf muscle, something that doesn't require precision. Now, a neuromuscular junction. Well, a junction is simply where two things meet. Neuromuscular is where a neuron and a muscle meet. All right, so we're going to be dealing with a motor neuron. Motor meaning movement. Okay, now if you remember back to the previous chapters where you talked about a neuron or nerve cell, okay, nerve cells have, or a neuron, have dendrites and axons. All right, we're just going to be dealing with the axon part of it. Okay, and there's only one per neuron. You'll deal with the other stuff later on in the course. So we have something called an axon terminal. Well, terminal means end. So this is the end of the axon that extends off the neuron. Now, it forms a junction with the sarcolemma, or the muscle cell membrane. Now, the sarcolemma has ACH receptors, which are acetylcholine receptors. And again, that's going to play in just a moment. The synaptic cleft is going to be the gap between the axon and the sarcolemma. Okay, so this and the bottom is showing the neuromuscular junction. At the top, you see the axon coming down, and then it branches into these axon terminals. And see how it's coming in contact with that sarcolemma as well. So if we move to the bottom picture, you can see inside that axon terminal, again, coming in contact with the sarcolemma. All right, so let's get into the nitty-gritty. You've got a nerve impulse is going to come down. Well, let's back it up all the way. All right, let's say you want to reach down, pick up a glass of water. How do we do that? It happens in milliseconds. So your brain had to send an impulse. Okay, that nerve impulse is going to release chemicals from the axon terminal. And the chemical that it releases is a neurotransmitter called acetylcholine. Now, acetylcholine is released, and then it travels across the synaptic cleft to the sarcolemma, where there are acetylcholine receptors. The receptors binding to that sarcolemma cause the sarcolemma to become more permeable to sodium ions. Sodium rushes into the muscle cell and generates an electrical condition called an action potential. Now, that's literally a nerve impulse running across the sarcolemma, and it travels down the entire sarcolemma. At the same time, potassium is moving out of the muscle cell. Now, that action potential travels down the sarcolemma and stimulates the SR, or the sarcoplasmic reticulum, to release that stored calcium. That calcium then triggers myosin to bind to actin, and you get the sliding filament theory. That's the myosin head attaching to actin. The sarcomeres will simultaneously pull actin toward the center, thus shortening or contracting the muscle. All right, so let's kind of look and walk our way through these pictures. All right, again, you see the axon on the top coming down and branching into the axon terminals. So if we see that picture a little blown up, that nerve impulse running down the axon caused acetylcholine to be released from the axon terminals. Now, you can actually see that in the picture. These little circular balls are called synaptic vesicles, and inside them, the little green dots are the acetylcholine. That nerve impulse came down, caused them to be released. They cross the synaptic cleft and bind to the acetylcholine receptors on the sarcolemma, which remember is the cell membrane. Now that action potential travels down the sarcolemma and then comes down and causes the SR, or sarcoplasmic reticulum, to release calcium. That calcium being released is then going to come down here and allow myosin to attach to actin and thus perform the sliding filament theory. In this illustration, you can see the sliding filament theory. Now, once that calcium from the SR came in, the myosin head was allowed to attach to the actin and the head cocked, thus using the ATP molecule. Now, here's this little arrow down here with the ATP. That's important because for the muscle to be able to continue contracting, it needs a new ATP molecule. So, myosin binds to actin, uses the ATP by cocking and pulling the muscle in. To let go and contract again, it needs a new ATP molecule. If there is a new ATP, then it just repeats the whole process over and over again. If there's not a new ATP molecule, then you end up with minor things like writer's cramp, where your hand cramps up because there's not enough ATP, or major things like rigor mortis. Okay, so you've got the contraction down. Now what about relaxation? Obviously, we want the muscle to relax, so how does that happen? Well, the action potential ends. Calcium ions then are reabsorbed back into the SR, or sarcoplasmic reticulum. The muscle cell relaxes and returns to normal length. Now, all or none principle. That simply means the muscle cell will fully contract or simply not at all. All right, sample question. Which ion is released from the sarcoplasmic reticulum and is needed for myofilaments to slide? The answer is C, calcium.